Our speaker today is Randall Wisser. He has done a PhD at Cornell University in plant breeding genetics. So he worked on disease resistance in maize. And after that, he did a postdoc at North Carolina State University in quantitative genetics. And recently, last year, he moved to Montpellier at the INRAE, so Institut National pour la Recherche Agronomique. What is the new E?
Environment.
And he's a research director for the Laboratory of Ecophysiology Plant Response to the Environment. And today he will do a presentation for us. So the floor is yours.
Okay.
Yes.
Can I take this? I keep this?
You can remove. Yeah.
Okay. Well, thank you all for coming today, and I especially want to thank Christian, who's been an incredible host. Took me out last night around the city, helped coordinate a last-minute visit here, and it's a great opportunity to visit, and I hope to get to meet some of the fellows here and the people here, even though I'll only be here for the full day today and tomorrow. So it's my pleasure to be able to share with you some of the work that we've been doing over the last, say, about 10 years. So really, I was a faculty member at University of Delaware in the US for about 10 years before coming to INRAE. Okay. Where starting to, at INRAE, we're interfacing some quantitative genetics and past work that I've done with ecophysiology and questions about exploring new diversity and things like this, and you'll get a sense of that as I go through the talk. So my title that I share with you today is on deconstructing and bypassing maladaptation to mind diversity, and I just want to start. Let's see if I can get this going by acknowledging a number of people that I've been fortunate to work with over the years. One of the fellows here is Seth Murray, so unfortunately he's at home. He's in the hotel today, but with us virtually. But in addition to him, a large group of investigators, Jim Holland, Nick Lauder, who unfortunately passed away last year, a young guy who passed away suddenly, so we miss him. But Sherry Flint Garcia, Natalia De Leon, Seth, and Wenwei Zhu at Texas A&M. So this group of investigators, we got together in 2009 and initiated a big project on trying to understand preadaptation in maize, and I'll talk about what that is. And there are a number of people I've highlighted in bold who are key to the particular work I'll share today: Juliana Teixeira, Heather Manching, Minjing Tian, John Doherty, Michael Dumas, Tekli Well-Decadin, Maria Lopez, Julia Winkler, Eric Shao, and a number of other people. It's a major effort, as you'll see, and the list should be much longer than is put here. And now at INRAE, I'm interfacing with a new group of people, and it's an exciting new adventure for me and hopefully for the group there too, including in particular, I mentioned François Tardu, Claude Welker, Alain Chapossé, Bertrand Muller, Pierre Matt, and Boris Parent, and a number of other colleagues at the LEPS unit. Maybe you know some of them, maybe not. But it's a great group of people, and in particular, it's a unit that focuses on the physiology of water deficit response, and particularly developments in phenomics to facilitate characterization of that response, and with high precision platforms and careful envirotyping of field trials. And one point I'd like to highlight is that there's abundant variation in the crops that we work with. I'm focused primarily in maize. It's part history and part its importance in breeding and agriculture, of course. But if we look at any scale of diversity or any scale of phenotype, you can find wide arrays of variation, whether it be the apical meristem measured here on the top left, whether it be disease resistance. If you look across inbred lines of maize, you will find very wide differences in disease resistance. So it's not this black and white differences of tall and short or resistant and not resistant. Instead, it's a spectrum of variation, quantitative variation that we find in abundance in maize and in other crops. It can be the length and size of the ears, the color of the seed, the plant architecture. And these are images here taken from this platform I mentioned. So you can just see the wide differences in variation for genotypes that come from Thailand, extracted from a landrace called Suwan, ones that come from lowland environments in Mexico and other places around the world. So you can find this wide array of diversity. And in the community of, I know this is a broad disciplinary venture at the BrIAS, so I should mention that the community of plant scientists have historically built up around the idea or the understanding that the phenotype is a product or a consequence of several components or sources of variation, including genotypic variation, environmental variation, the interaction of these two things, in addition to some leftover unexplained variability. And over the decades, there have been great advances, and especially in recent years, for tools for measuring this phenotypic variation, using experimental designs as a foundation to separate the sources of variation systematically, and then a plethora of methods for dissecting the underlying basis of these different components of variation, and now more recently a large focus on algorithms for predicting phenotypes, not at various field scales. So despite this very huge reservoir of allelic diversity, the history of breeding, the history of domestication, breeding, selection, that entire history has sort of whittled away a great deal of that variation that exists still today, but it's underutilized. So if we look at modern varieties, there's you get this little funnel perspective of you have some number of alleles, and through the processes of bottlenecking and selection, you lose some along the way, and consequently you're stuck with a smaller pool of diversity, and it's that which particularly elite breeding material is constructed around. And so if you look at modern varieties or elite varieties of crops, you'll find that they represent a relatively narrow slice of the amount of variation out there. We can talk about elite maize and consider and know that it constitutes less than 1% of the global populations if we just think about populations as a thing. And they capture less than half of the variation, about 40% of the genomic variation, SNP level genomic variation that's available more broadly. And so really the basis for the things we're doing is to say, if we think about transformation of agro-ecosystems, if we think about battling issues with climate change, if we think about an array of issues that we try to deal with, the argument I'm making is that without an influx, not alone I'm making this, but the whole community would make this argument, without an influx of new diversity, there's quite limited scope for adapting crops to these different kinds of challenges. We need the diversity to achieve that. And some good examples, if you look back at exotic material that's not widely incorporated into modern material, you can find all kinds of interesting climate adaptations. And here's an interesting one from a wild strain of maize, actually, that is a modified stem structure that gives rise to waterlogging resistance or tolerance. And in another case, a publication recently highlighted a particular landrace, although it's not restricted to this particular landrace, of maize that forms a thick mucilage on aerial roots, and in that mucilage, bacterial communities can form and fix atmospheric nitrogen that's incorporated for plant growth. And so in that case, it was an environment that had under low nitrogen conditions. So it's this other type of adaptive mechanism. And there are, we can keep going and going, but most of the material that's out there that's exotic is generally undercharacterized. We still are only scratching the surface of what that variation looks like, what it has to offer, etc. Now, when we could talk about moving into the temperate zone where European, North American breeding, we have a big issue with trying to characterize that diversity. And that issue is primarily one of several issues, but this issue is the issue I'll talk about is a big issue, is that when you try to grow material that's adapted to some, say, tropical environment in France or Belgium or whatever, so temperate zone, then we run into a problem of maladaptation. The material is just so poorly adapted to that environment that it has all kinds of growth abnormalities. And one of those abnormalities is highlighted here in these women at a school in Iowa in the United States that stack themselves up on top of ladders to compare how many people it would take to get as tall as that corn got in that environment. And the reason why this corn plant got this tall, which is excessively tall, meaning it also flowers very, very late, if at all, is one of the big factors with that is photoperiod or sensitivity to photoperiod. So you know in the summer we have long days in the temperate zone, and those long days cause plants to get stuck in transitioning from vegetative to reproductive growth. And so while they're stuck in that non-transitional phase, they just accumulate more and more biomass and sometimes don't flower. And if you don't flower, you can't cross it. If you're very big and so you can't evaluate yield in that environment, you can forget about that typically. And then other traits just tend to be confounded by this issue of maladaptation. So you kind of get a skewed perspective on the merit of those materials when you grow them in your sort of target locations. So fortunately, simple breeding can overcome this problem. And this is a great picture. I love this picture that it's actually from a field that we had in Delaware, and it's from a breeder originally developed this population in Iowa. It's Arnold Hallauer. Through his breeding program, he took tropical strain of maize, a lowland tropical strain of maize, as a population, and over 10 generations, gradually moved its flowering time from extremely late tall plants, as are shown here as examples in generation 0, to by generation 10, plants that were the norm for the temperate zone of region of the US Corn Belt. And so in this case, what's shown are two rows side by side from the same generation for generation 0, 2, 4, 6, 8, 10. And it's grown in the same environment. So here, immediately, you can see evolution in action. You make a direct observation from starting with one population and over the course of selection by breeders for 10 generations, because we have no environment confounders, you can see the direct product of that selection process. And it's a fascinating transition from the morphology and size and as a number of characteristics a suite of characteristics that are shifted as selection for flowering time is adjusted. So the problem, of course, is that I just said it took 10 generations, and 10 generations in Iowa means you're breeding in summer environments. You have to wait once every summer, and that takes 10 years. So we have this major phenological barrier to accessing that diversity I mentioned. And then the other problem becomes, okay, you can do this for one population, but that's already a big task. But if there are thousands of populations out there, and which ones should we choose? Which ones are the most likely to give us the useful adaptations if we go through this process? Because we can't get it right now. We can't get around this process of needing to adapt it. And so there are other open questions about how do we really incorporate new diversity. And we'll talk. We're trying to think about that too. So it's nice that we're working on flowering time. As many of you probably know, it's very deeply studied. And in maize, there's been some great advances. And from that work, we know that flowering time, like all the other traits I mentioned, is sort of typical for traits in maize and other crops, is that it's quantitative. And now we know a lot about the quantitative genetic basis of that variation. And so on the one hand, when you look across multiple populations. This is a collection of 25 separate populations that were dissected for the QTL that segregated in those different populations. You find that taken together, there are about 30 loci that are detectable by statistical techniques that are positioned across the genome. So that those 30 different regions, plus actually many others that have small undetectable effects, give rise to that variation. And you can just think about if you had 30 die, how many combinations you would get every time you rolled made a new roll. You would get tremendous numbers of combinations. So we can already imagine how you can give rise to the kinds of variation we see in ourselves and we see in plants, et cetera, for quantitative traits just by the sheer number of loci. But it gets more interesting because there are actually multiple alleles too. At a single position in the genome, you have alleles of different sized effects. And so you can take those 30 loci and then pile on multiple alleles and you get more and more combinations and more variability. But the other point to make here is that those effects are very, very small. So any one particular allele only is contributing to a difference in flowering time of like 1 day. And those are big effects. So, people were talking about a lot about potential for even transformation and CRISPR activities. I mean, we have problems of just figuring out what the causal variants are, and then whether those variants can be manipulated in ways that can have a really meaningful shift in the phenotype. And so that's important to keep in mind as we try to tackle these quantitative traits in different ways. And then also, we know a lot about the environment effects. We've learned, I mentioned already in case of flowering time, we know it's controlled a lot it can be controlled by photoperiod if there's photoperiod sensitivity. Temperature is another important cue, water. So there's different environmental variables. And here, the graph just kind of shows that the intensity of the color indicates where that allele is expressed in a particular environment. So you can see that there's a lot of progress in dissecting the phenotype through these methods that are developed for many over a long period of time. Where we've been trying to work now is trying to understand more about the interaction of genetics, environment, and selection, because we're adding that component of selection for preadaptation and trying to understand the genetic basis or other bases for that phenotypic change that I showed you in that one figure when we started. And I'm not going to go into this this component at length, but there's a lot of things going on now today with all the advances that I mentioned at the beginning, especially in microbes, for studying this interaction. And they coined it as experimental evolution. And there's all kinds of fascinating studies. There's a group at Michigan State led by this Professor Lansky who showed after about 50,000 generations of selection that bacteria have accumulated a new ability to use a totally different carbon source for survival and fitness. And so from these experiments, we're learning about the role of mutation, the impact of epistasis or interactions between loci, as well as the role of standing variation, the existing variation that we're starting from, and how those are contributing to the way that phenotypes change. And in our case, it's sort of experimental evolution. I sometimes feel inappropriate to call it evolution just because it's such short scale process and it's very artificial in the context of breeding, but we can maybe call it experimental breeding. So we've been using this experimental breeding strategy and trying to move to a particular target. And as I mentioned, that's early flowering time. So if we take some exotic material and we breed it to a particular flowering time, so it's adapted to our location, we want to know a lot about the genetic basis and how of that change. And so this was the project I mentioned that we started in 2009, and we spent a lot of time, actually. It was very difficult developing new germplasm and some tools and techniques to handle the type of material we were working with. They're not inbred lines, they're populations, they're heterogeneous, heterozygous. So that created a number of challenges, even for genomic approaches that were existing at the time and even still today. So, and then the major component, the major element was to dissect the phenotypic basis of that change in flowering time. And then now we have a new project where we've been advancing a genomic selection technique. So it's basically a prediction method that allows us in off-season nurseries where we're not in the environment, the say light-induced environment, long day environment, to actually make gains in selection. And so for the selection experiment that I mentioned, we did something we call parallel selection. And so what we did first is we created this all tropical synthetic, and these parent lines were chosen specifically because of some agronomic characteristics, and because they were tropical and photoperiod sensitive and temperature, not temperature adapted, but basically late in the temperate zone. So we had this systematic crossing scheme. We created this single point population, and that population was distributed to 8 locations throughout the US across a latitudinal transect that was about 3,100 kilometers in range, going from Wisconsin down to the island of Puerto Rico. And so the slide over here shows the points at which we had populations grown. So starting from a common base population, we call it generation 0. We grew out the 10,000 plants, and at each location, we selected the earliest 500 plants, and then advanced them, crossed them in a systematic way at every location, and advanced them to generation 1, and repeated that process using the location-specific selection. So you kind of have these lineages, these location-specific lineages that come out of the experiment. So with that said, I want to point out a feature that we've seen in some other, from another material that had underwent the selection. I mentioned at the beginning, this population underwent a decade of selection. So in a study that where we took that population and grew it out at those other 8 locations, we looked at how the flowering time changed, not only in the place where it was originally selected, but also at all of the locations where we were performing selection with this new population. And that's what this figure shows. You have the y-axis showing you the flowering time going just in simple days from 50 to 100. And then on the x-axis for each environment, just a 2-letter code for the states in the US, Wisconsin, Iowa, Delaware. So latitude is decreasing going that way. Day lengths are getting shorter going that way. Temperatures are getting hotter going that way. Nighttime temperature, let's say. You have the generations 0, 2, 4, 6, 8, 10 at each place. And so the thing I'd point out from this is to note the sort of very strong change in flowering time. You can see the population started. I lost my pointer. Sorry, I'm using something that's not working for you to see. Population started at about 95, 100 days to flowering, and was shifted to about 870 some odd days to flowering. So a really big shift in flowering time. And you can see, if you look across all locations, you get the same response. So it doesn't matter if I grow that population, even though it was selected in Iowa, if I grow it in Puerto Rico, it caused it to show a similar change. Generation 0 is later than generation 2, which is later than generation 4, blah, 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 et cetera. So this selection in one location creates a sort of phenotypic convergence or parallelism across all locations. And you can see, if you look carefully, that the slope of that response does differ. It gets a little bit less as you go into the southern locations. It's greater in the specific selection environment, and latitude's very close to it. And you can also look, if you try to imagine the average among all the points, and you go across all locations, you see that the absolute days to flowering is also changing a lot. And that's an environment effect. So when we can decompose the sources of variation, like I mentioned in the beginning, and when we do that for this population, we see we have very wide phenotypic variation. We have effects that are genetic, that are quite broad. They span across the whole all the locations. The same population grown at all these locations shows the same response. There isn't like a crossover between the directions of selection in 2 different locations. The environment effect is actually bigger when we estimate it. It has a bigger overall effect, or captures more variation of the phenotypic differences. And the interaction between genotype and environment is quite small. And we have very little leftover residual. So we're explaining most of the variability and finding that it distributes across the sources in this particular way. So, and I should mention the G by E, if there was strong G by E, that would suggest there is very local effect. So we see a minimal component of local effects, that is local adaptation, let's call it, selection for early flowering time, but local adaptation, and a bigger component of broad adaptation. So what this implies to us, or maybe when you first look at it, I don't know, our first thought is that the expectation with something like this is that you then have a common architecture, a common genetic basis would describe the reason why that population looks the same in all these different environments in terms of the relative change across generations. So in our other experiment, the parallel selection, mentioned we use a standardized selection protocol. And to look at this question of this phenotypic convergence or parallelism across environments, we had a sampling strategy and a technique that's called extreme mapping. And what we actually did in the times that we grew out those 10,000 plants, we didn't only select the earliest 500, but we also genotyped, sampled tissue. We actually sampled 500 tissue from the earliest and latest tails and 1,000 random tissue from the population itself. And it resulted in a 32,000 tissue collection sample that we have stored and kept away. And then we genotyped 384 early and late tails from each of the locations. And there were 2 generations of selection. So we ended up genotyping about 12,000 samples in that. And we had about 150K SNPs in these 16 contrasting cohorts. So we have generation 1, taking the 0, taking the early and the late, and you have that across 8 locations. And then you advance a generation with the lineages, and we repeated that across the locations. So we have these 16 contrasting cohorts. So the idea is if I compare the allele frequencies at any one position in the genome between the early and the late, I can use it as a basis for detecting whether there is a significant difference in the frequencies of the alleles, and by doing that, I can locate the things that are underlying the phenotypic difference and driving the change, presumably. So indeed, applying this extreme mapping technique, we find about 11,500 flowering time SNP associations. 75% of those end up being on 3 chromosomes. And we find that because of the knowledge of flowering time, so I can try to describe this better. We have the chromosomes listed here, the number of markers that were detected to be significant, and the proportion of the significant markers across as a relative basis. And then each of these graphs shows the chromosomes. It's a complicated graph, but it's there's sticks that are highlighting the locations of particular genes that we know, and then there's thick lines, these black and green lines that are indicating the flowering time SNP associations we detect in generation 0 or generation 1. And then, so we find that we have associations with numerous known flowering time genes, and of course, always these studies turn up things we didn't know before too, so those are all in there as well. And we find that they're both generation-specific and non-specific associations. So that means that we detect some things in one generation that aren't detected in the next. It's not like the same genes are being detected. And let's not forget that phenotypically, if I grew them out, even though these differences exist, you still phenotypically see that these populations have moved in the same direction in different environments. And the flowering time SNPs are clustered at times across very large sections of the chromosome. So here, I really highlight this on chromosome 10. These black SNPs are all showing a significant association across the entire length of the genome. So we'll come back to that probably. I gotta watch my time, I'm going slow. So, so we can take a look at the extent of sharedness between SNPs that are detected to be associated with the difference in flowering time across all these cohort pairs. And the upper graph here is showing for generation 0. It's just to give you the idea here that we have some degree of sharing between certain locations. So there's location one on the left, the low latitude location, to the high latitude, so each points to the location. And then these sets of locations have a higher degree of sharing of the same SNPs being detected across those among those locations, those different lineages, than the southern locations. And then by the time we get to generation 1, all the things we're detecting tend to be not shared. They're different sets of things, different genes or alleles that are being found. So there's not really this like universal architecture. There seems to be something more complicated going on. And so you can ask the question, how do we reconcile this? How can we have different sets of genes being selected in one environment, and then when we grow that population in another environment, it shows the response that we saw in the other environment? So it's just a little confusing. So the way we approach this is we did this clustering technique where we basically try to ask which SNPs show the same, more similar magnitude of a difference in allele frequency across the locations. It's kind of like a profile, imagining a profile. So we go to generation 1 and we look at the allele frequency differences between the extremes for all locations, and you can imagine that forms some kind of profile. And then we can look for all the SNPs and see which ones have similar profiles and different profiles. In doing that, we uncover some features about what's going on at the architectural level. And so one of the things we can see is that, and I haven't shown you the clustering yet. So what this is showing you are the effects on chromosome 10. You remember that big set of SNPs that was selected. So we can see going across chromosome 10, this is chromosome 1, 2, 3, across 10 for these particular environments, they all show some SNP effects, but a lot of those SNPs are not exactly the same ones. They're not shared. So it has a similar architecture, but they're not exactly the same SNPs. So the whole section of the chromosome shows up, but different SNPs are getting tagging that outcome. So that structure of architecture is more similar if you look carefully among, not so carefully here, but among these northern latitude locations, and then these southern latitude locations tend to have some degrees of similarity as well. But between this mark of what is central Texas and North Texas, so this is where Seth is at, and then North Texas is his colleague north of him, you just have this kind of dividing line there, and it turns out that that's a photoperiod sensitivity. So to do this in a sort of deeper way genetically, because we have knowledge about the parents, we can do some kind of parent tracing across these generations of selection. And so what we had to do for that, we had to develop some techniques or adapt some techniques that were available. And so what we had was SNP data. This is what SNP data would look like. You have, for any one sample, you have a chromosome 10, and each vertical line is showing a marker position. And each of those marker positions, you have a variant of the nucleotide. But what we did is we have an algorithm that will go from the SNP data with input about the parents and convert it into what are called haplotype maps. And so now I know for this same one sample that this blue section, this section of one of the two homologous chromosomes inherited the haplotype from this parent CML373. And the color scheme kind of continues like that. And so we have these haplotype maps for 12,000 samples. And we use that to try to analyze the data and get a better understanding of what's going on. And so one of the things we see when we look at the parental haplotype, so I have the SNP in black, how the frequency difference between late and early differs, just talking now about generation 0, not forgetting about the next generation. You can see the black line showing you the SNP difference between early and late tails, and then now we can parse that out into all the parental haplotypes. And then you see that there's basically no difference between the allele frequencies of the parent haplotypes at these southern locations, and at the increasing latitudes, you see this shift where some parent haplotypes are really favored, and it's consistent across the locations. So basically what we see going on with CML341's haplotype is showing up in 1, 2, 3, 4, 5 locations as the favored allele or the unfavored allele. It's being deselected. And the other interesting thing when you look at this, just for more genetics point of view, is that there is a haplotype series or an allelic series. So there's a very clear differentiation between the green, the yellow, and orange. And so already you can see at this particular position I'm showing you that there are a multitude of alleles, not just different loci. And it turns out that this particular thing I'm showing you is associated with an extremely large effect. From statistical terms, it's enormous. Has a negative log p-value of 120. And it's centered on a well-known gene associated with photoperiod that we knew from the beginning was segregating among the parents of the population. And it exactly matches with our expectation of which parents contain the particular causal variant are showing deselection, and the effect is located at that position. But why is, if I go back, it's not so easy to see. But if you look, it's really like the effect is spanning the entire thickness of the chromosome. And so why is that? So it turns out that's an issue with limited recombination on chromosome 10. So on that, so this is a figure that shows that same sort of, don't worry, don't get lost in the detail here. I don't want to, this can be an overwhelming one, but what it's showing is when we go across the chromosome now, at one location, one of those northern latitude locations, we can see that parental haplotype of CML341 is the favored haplotype, the unselected haplotype across the whole length of the chromosome. So it's not just pinpointed at the position we know where ZMCCT10 is at. It's actually dragging that whole entire haplotype block from that parent out of the population. And we know now that there's just very limited recombination, which is probably a property of the parental lines that are used. So something is not allowing the chromosomes to recombine, and that basically creates a situation where you select on one major effect thing, and even if there's small effect things nearby, they just get dragged out. So maybe there's excellent tolerance for drought here and here and here and here, and nitrogen use efficiency across that haplotype. Well, it will be gone. By the time we do the selection in the temperate environment for early flowering, we'll have lost that variation if that variation were potentially important. So that's sort of maybe a unique case, we don't know, but it's something that we see when we go from the SNP scale to the haplotype scale. When you look at the SNP scale, you're not seeing that type of that same level of reduction. So, but in contrast to this sort of one major locus, there are many, many others with the same sign effect, but they're too small to detect. They're detected sometimes in one environment and not another, but when they're found in other environments, they have the same sign. So the effect of the allele is the same in Iowa and Puerto Rico, but in Puerto Rico, it's just a little too small to pass our threshold, and in Iowa, it's just big enough to detect it, and that's why it shows up as a significant SNP in the first place. So there's many of these things with many others with same sign effect and too small. So we think that there's this two component thing going on. We have this locally large, that is locally meaning it's associated with a particular set of locations, and it has a large effect, and widely small. There's many, many other things that are have a wide effect. They operate across many environments, but they're very small. And when we look at the relationship across all SNPs in the genome, their effect estimates, we do see exactly that scenario play out. So what you have are alleles that are specific to one environment, and that's why they have a big allele effect on the y-axis going up that point. And some alleles that have a big effect in environment B, but you have many, many small ones that give you some partial correlation. And so what we think is there are a few locally large and many widely small. This combination could help to explain that parallelism, but it's probably not the full story. And the other thing I should mention here, just to take note of, is, I'll just reiterate this in a second, is that when we look at this position and this position of these effects, note that they almost have zero effect in environment B. So the big things that have the things that have a big effect in one environment have a negligible or no effect in another environment. And And so that's sort of the combination of features that are happening here. We see few locally large flowering time alleles that are really specific to particular latitudes. And these have negligible effects in other environments. So there's kind of no trade-off in terms of flowering time, at least. And there are many very widely small flowering time alleles spread all across the genome. And this gets a little more into that combinational, those roles you get. If you have a lot of possible combinations, you just can imagine that if you add those separate roles up, sometimes you'll get exactly the same sum. And so consequently, you can actually have something called genetic redundancy in this system where you have many loci with small effects, where you can get lots of combinations. Those combinations can lead to the same phenotypic outcome. So, and there's strong evidence for this in some of those experimental evolution studies I mentioned at the beginning. So we think that that's probably also a factor. So there may be things that are widely effective across environments, but it may not even be the same set that is getting selected in one environment and another. Nevertheless, it still works when you take it into a separate environment. So, so you get into this sort of complex situation. It's not sort of the simple story of genetic, that you get this loss of diversity under selection. You actually have a very complex architecture, and that architecture can generate genetic redundancy. Like I mentioned, can actually be a source of maintaining variation. This could help explain, if not mutational variance, how standing variation itself can fuel very long-term evolutionary change. And this comes from also evidence from another population that we looked at, where the history of the population was very different. So essentially the demography of the founding population that you start with is a very strong influencer on the outcomes you see in this process, the following process of selection. So in one population, we found that maize can get to that adapted state without losing almost any SNP diversity. Everything's still there. And in another, we find that recombination, at least on some chromosomes, it seems to be really driving stuff. And if it's driving stuff out, it also, if you're not recombining, you're not making new allele combinations, you're not allowing the gains for selection to go as quickly as they could. So you're also constraining the response or phenotypic change that could otherwise be if recombination allowed those new combinations to form. Importantly, we know, it's been shown by others and ourselves now, that tropical maize or other exotic maize populations can be adapted for flowering time to the temperate zone from standing variation. Maybe mutation is playing a role, it's possible, but certainly we have enough evidence to show that the standing variation itself can allow for that. So with that said, we also found in our work that this has some negligible effects of the environment-specific flowering time loci. So I could enrich it in one location and expect that, at least from flowering time point of view, it has no impact in another location. So with that, plus this polygenic system, you can use that, you can think about that in a genomic selection scenario to help drive more quickly the phenotypic change. And so we have a project, just have a few more slides left, maybe 3 more slides. So we have a project now on speeding up preadaptation. This is in its 2nd year. And the idea here, and this originated in the US before I moved here, is that there's an adaptation barrier that's driven primarily by the latitude, as I just highlighted. And so you have short day and long day, and then you have collections, whether it be the National Germplasm Bank or whatever collection it is, or land races ex situ, and you want to move them into the temperate zone. Then we have a scheme highlighted in green here, where we basically grow the population, those 10,000 individuals, like I said, and we randomly phenotype a group of individuals, and we use that to train a model that will predict the early types. And then we apply phenotypic selection in that location, so we select the earliest ones. Then we move the population to a southern location in the winter season of the temperate zone. So you're in this, and there we implement genomic selection. So now what's happening is all the things that are latitude specific, environment specific up here can be selected in that location. You don't need the inducer You have the genomic selection technique to tell you what should be early back in that temperate zone. So you don't need that environmental inducer. It's like selecting for disease resistance without disease being present. It's like the same type of concept. So that's a strategy, and we've made it through some intensive genomic selection stuff. In practice is always more complicated than in theory, but we managed to get that done. And so Nicole Choquette is a student, PhD student at North Carolina State. And in this work, we showed that phenotypic selection alone can achieve about a 2-day shift per generation. And it takes, as I said, you have to wait every summer. So now by just adding in that extra off-season step, we can get to the point of an adapted state in not quite half, but nearly half the time it takes to do by phenotypic selection alone. So that's a major, that could be quite good for bringing in stuff once we know what we want. I had these more as like closing points. So now I'm at the LEPS in the INRAE, and there we're trying to take that history of the work we did in quantitative genetics and trying to understand adaptation for early flowering and combine that with a venture to try to explore the diversity and figure out other ways to kind of bypass photoperiod. So we have a new PhD student in the lab, Justine Drought, who is working on this question of whether we can phenotype in platforms for water deficit response in short day time periods in controlled systems, and whether that can give us meaningful information about what the material should be characterized. And she's doing some other really interesting things with photoperiod that I'll save for another time, or we can talk about more. But the basic driver of what we're doing is which genotypes are at the right place in the phenotypic space. And so we're using these platforms and advances that are founded on 20 years of ecophysiological developments that have led and developed new phenotyping systems with an emphasis at the LEPS and drought and heat. And we're combining this with some virtual methods that we can discuss. And then, of course, we have to apply proof of concept and validation and try to figure out if these virtual techniques are working. And so I can close with this. This actually gives a better summary of the current group at the LEPS. And with me, I have 2 postdocs right now and a PhD, and we're actually searching for Emily Millet. She recently got a permanent post at INRAE, so we have an opening for a postdoc to take her position. It's an exciting project where we're partnering with INRAE, a mathematics group, and they're called the Data Shape Group, and some ecologists, and trying to explore how we can look at plant variation in crops in the ways that mathematicians and ecologists think about things. So we're really focused on trying to understand the phenotypic space in terms of, and that is the multivariate space that the varieties of tropical or temperate origin occupy and how they compare to one another. So she's working on that. And then we have BIPASS, which is Justine, I mentioned already a bit, and a virtual genotype to phenotype project by Arnaud. And he's working on integrating genome simulation, whole genome simulation with crop growth models. And so in that way, we have populations that we now are gathering enough empirical information to parameterize a simulator, a genome simulator that can create new recombinants, and then feed those recombinants, the phenotypes of those recombinants, into a crop growth model and test the performance of different ideas for breeding in different field situations. And with that, thank you. Happy to take questions.